Hi, I'm Akriti Kapoor. I'm a second year Applied Biosciences Professional Science Master's student in the Diagnostic Laboratory track. I've been working in Dr. Nelson's laboratory in the Pathology department. Nelson Lab has been working on a very rare neurodegenerative disorder, which is known as the TAF1 syndrome. It's an X-linked disorder and it occurs in children. Today, I'm here to present the implications of TAF1 deficiency on cell cycle progression and proliferation. To give you a little bit of background, cerebellar ataxias are a group of neurodegenerative disorders which are characterized by motor dysfunction, balance problems, limb and gait abnormalities due to cerebellar atrophy. Our lab has been performing clinical exome sequencing, whole genome sequencing, messenger RNA sequencing over a period of 2 years on a family trio containing the affected proband, his unaffected parents, and grandfather. The 3-year-old male proband presented with global developmental delay, motor, cognitive, and speech, hypotonia, and possibly ataxia. Also, an abnormal MRI with progressive cerebellar atrophy of unknown origin was found. The proband has a variant of TAF1. Now, TAF1 is a very important gene since it's involved with gene transcription in most of the cells of the eukaryotes, and how these variants of TAF1 lead to neurological phenotypes is largely unknown. Our lab also demonstrated that the variant or the mutation in TAF1 is present in the second bromodomain. Now, bromodomain is a 110 amino acid long protein domain that recognizes the acetylated lysine residues on histones. Previous literature has also shown that TAF1 mutations are linked to abnormalities in the cell cycle, and we postulate that variations or mutations in TAF1 can alter cyclin expression, which can ultimately lead to disturbances in the cell cycle. To address this hypothesis, I wanted to study the effect of TAF1 depletion in human neuroblastoma cell line, IMR32 cells, and how the effect actually impacts cell cycle progression and proliferation. Also, another aim of the study was to assess cyclin D1 and A2 gene expression through RT-PCR in TAF1 deficient cells. So IMR32 cells, they were transfected with siRNA duplexes to do TAF1 knockdown for 2 time points, 24 hours and 48 hours. And as the first diagram, it shows that RT-PCR result analysis showed there was significant amount of TAF1 depletion in both the time points compared to the control. But when effect of TAF1 depletion was studied on cell cycle progression, there was any effect shown by flow cytometry. Similarly, there was no effect on cyclin D1 and A2 gene expression compared to the control in TAF1 deficient cells as shown by RT-PCR analysis. So to conclude, we can say that more TAF1 depletion studies need to be done with different cell lines like HeLa cells and TS13 hamster cell lines to validate our results, or to see if this occurrence is common across cell lines or if it's only restricted to IMR32 cells. And if you have any more questions regarding our research, feel free to ask and feel free to go over our poster. Thank you.